and he'll be talking to us about motif enrichment in regulatory regions.
Shall I clap it down? Yeah, thank you. Hello, good morning, everybody. And I'm really thankful for the organizer for this opportunity to speak about my work. And so I can skip some concept introduction since we're already seen before. So just to say that we have seen transcription factor specificity is crucial for gene regulation with other layer of regulation. And in the last decades, a lot of effort has been done to develop technique to clarify and quantify the specificity with in vitro models. And our lab proposed the protein binding microarray and developed it, in which the oligonucleotide of a microarray are double-stranded and then assayed with a fluorescently labeled transcription factor. And then this can, so if the design is properly done on the microarray, we can score all the possible 8-mers in the 8-mer space through an enrichment score, and so basically arrive to the most recognized 8-mer sequence by each transcription factor. And by doing that, and here on the right you see that the score that we typically use arrive up to 0.5. So then this data can be processed in different methods to create a specificity model as the position weight matrix that we have seen before. And this is crucial to then go to regulatory regions from genomic data and scan for motif enrichment and predict transcription factor bindings. And now there is several genomic data that can be interested from this approach. And protein binding microarray has inspired the previous DREAM challenge in DREAM 5. And the main question there was which is the best algorithm that can basically recapitulate the whole pipeline, and even which is also the best model to catch the specificity of transcription factors. And one of the, several questions were answered by the challenge one that was that in general for, let's say at that point, for that survive 90% of the transcription factor, the position weight matrix model are a quite competent and effective model. But still, some questions were unresolved, the fact that the best pipeline is transcription factor dependent. So different transcription factor can have better description with different algorithm for the construction of the position weight matrix. And in a, let's say, important fraction of the transcription factor repertoire, position weight matrix have a big limitation and cannot really catch the specificity, and in that case, K-mer model are definitely were proposed as much more effective. And so to address these issues and to gain some further understanding on this process, we developed this let's say different approach that was more a K-mer-based approach. So we imagined to try to use the more specific K-mer, 8-mer for a transcription factor recapitulated in vivo by protein binding microarray, and exploit the fact that typically transcription factor from the same family should basically exhibit the same specificity to the same regions. And if we can compress this and in a sort of module that link a subset of transcription factor, we can then, by systematically applying this to the surveyed transcription factor classes, create a sort of minimal glossary for the transcription factor family specificity. And the idea was then to apply that to the, let's say, the number of survey and data that have been produced in the last years. So we took 640 non-redundant transcription factor for metazoan, and using the two main data banks for protein binding microarray like UniProbe and CSBP, we added 70 new data from our lab, so covering in total more than 25 transcription factor families. And then we applied the following workflow. So at first we ordered the 8-mers through a hierarchical clustering, then through a dynamic tree cut of so a fixed size, we clustered them, and we associated to each cluster the most recognized transcription factor. Then we compare the different kind of size for the dynamic tree cut. So, and we merged the clusters in what we call the preliminary modules. Then by removing the unclustered 8-mer transcription factor pairs, and we reclustered then the data set, and we arrived at the end of the day to this sort of heat map where we had 114 glossary modules, and that are basically the island within the boxes, and every box you see is capturing a sort of tight specificity region. And with that, we covered like 30% of the total 8-mer space, and we were able to maintain roughly more than 80% of the initial transcription factors. And if we look at what every modules contain in terms of transcription factor, we see that more than 85% of them just contain transcription factor for one single family. And more precisely, when we zoom into this, what we call pure cluster, we see that there are different hierarchy of transcription factor aggregates. So sometimes you can just recapitulate an entire transcription factor family, like in the case of PO, sometimes subfamily, like in the case of homeodomains, we have a dozen of transcription factor subfamily, and sometimes even single transcription factor that have a sort of divergent specificity for their class, like FOXM1 from the forked transcription factors. And then we use this glossary as a tool for motif enrichment in genomic data. So we started with Chipseq data, and there we recognize that there are two main scenarios that we have to deal with. In one case, we can imagine that the motif predicted by the module is the same as the motif expected for the Chipseq, because there is a direct binding of the transcription factor chipped in the assay. Second kind of scenario is when the chipped transcription factor, transcription factor 1, is related to a motif through another transcription factor, and so there is an unmatched pair between the motif of the module and the motif of the Chipseq transcription, the Chipseq transcription factors. So just to start with the first one, we took 241 site-specific transcription factor from ENCODE, and we saw in the specific module, we compare the IUROC enrichment of the modules versus of the more proxy position weight matrix. And this followed a little bit analysis already presented in previous work, also in DREAM5. And if we do that, we see that in most of the cases, that 77% highlighted, the two models, so 8-mer and PWM, are agreeing. So there is a general similarity in predicting enrichment, but in a substantial subset, the 8-mer models were able to see the enrichment that it was expected. And you see here in the bottom right quadrants. So, this is not the case in the case of PWM, that were not able to outperform 8-mer model in the kind of prediction. So, suggesting or like indicating that the modules in a fraction of transcription factor have a higher sensitivity for direct binding, in agreement with previous results. And then we move to the second scenario, where let's say, unpredicted transcription factor is bridging an interaction between like our chipped peaks. And in this case, so we compared the peaks with the remaining modules that are not expected to be present. So having more than 25,000 enrichment comparison. And in this case, what we saw that most of the time, this is a rare event, and so there is a rare regulatory event that is not predicted by both of the modules, by both the modules and the position weight matrices. But what was quite interesting is that in this case, 7% of the pairs were predicted by the PWMs. And since it's such a rare event, this seems to suggest that the 8-mer modules are more specific in sorting out this false positive, although this is not really so ruled out by this analysis. So we try to look more inside the data, and here an example, like in the case of the POW module, here there is all the Chipseq module pair enrichment in this, and they are ordered according to cell lines. And what you see is that the three enrichment are predicted all present in embryonic cell, in embryonic stem cells, and this is quite well pinpointed by the fact that the only POW factor that is upregulated is in this cell line is OCT4 in embryonic stem cells. While when we do the same enrichment analysis with a collection of PWM for POW factors from different techniques and different models, what we see is that most of the time we see enrichment in cell lines where the RNA-seq data, so the expression does not support the presence of a mediating transcription factor. So, then we moved to a second example. In this case, we wanted to see whether we can recapitulate motif enrichment in open regions. So we look at 10 cell lines where we had both DNA-seq and RNA-seq data from ENCODE. And also in this case, we see that for the most enriched transcription factor modules, we have we saw the clear enrichment pattern, and this pattern were really different between every cell lines. But in what was quite interesting, that if we like zoom in each of them, like in KLF, and we match this with RNA-seq data, we see that typically just one transcription factor is popping up as sort of responsible for this opening of the chromatin, and the one that is expected sometimes is well established, like KLF1 in the case of erythrocytes. Sometimes it's just, we have found that it's just reported, so we corroborate previous finding, and like in KLF5 for cervical cancer, while in several cases we see novel candidates like SP5 in hepatocytes, so that we may suggest as further studies. So in summary, what I presented you today is this glossary, minimal glossary for 8 mer modules specific to transcription factor families and some families, and this is based on in vitro PBM data. And the modules, in agreement with DREAM5 challenge, outperform position weight matrix in motif enrichment, and especially they are both more specific and more sensitive for the different kind of bindings. And third things, this module can be also really interesting in going to DNA accessible region where you don't have a priori idea of which transcription factor you would like, so you will find. So like, in general, we think that this could be a sort of compact and rapid search tool for having a first overview on genomic regions, and since is quite, so the modules are quite compact, this can be also provided as an online tool for enrichment analysis. Okay, acknowledgement, I would like to acknowledge Marta Bulik's lab, and especially Marta, that has been really supportive, and Luis Barrera, ex-PhD, now working at Editas, that has really shared a lot of discussion with me, and I'm ready for your questions.
Thank you. So one of the places where we see a big difference between PWMs and 8-mers is CTCF.
Yeah.
Is that clear from your comparison plot?
Yeah, but CTCF is not part because it's a long motif. Our motif, the motif that we can typically recapitulate with the cluster are 5, 6. The length is 5, 6, 7 nucleotides. So CTCF is not. No. So like P53, CTCF are too long, so they are not catched by position, by a protein binding microarray, by construction.
So there's something I might have missed, but I didn't really get how you compute the motif enrichment. how do you do the enrichment analysis? How do you assess the enrichment of a motif within a specific region or within?
In Chipseq, you mean?
Yeah, for instance, yes.
I take the best 500. Okay, the data are these narrow peaked from ENCODE. Then I take the best 500 peaks, and then for the motif, I look if one of the 8mers present in the module is present, at least one is present in the peak, and then I move the E-score threshold to see to basically to have a sensitivity over different E-score thresholds to see, and this created the IU-ROC curve.
So you're not using any background at all? When you're not using any background
the background is I compare this with the denucleotide reshuffling of the peaks, and this is like what seems now. There is both, as you mentioned before, this keeping the GC content and the same length, the same kind of feature. What I tried both. I tried three things, basically going just in the flanking regions, maintaining GC content, so the features. Also, this becomes really computationally difficult, and the denucleotides seems to have the more clean outcome, because sometimes you have also other features like some repeating GC, AT, that can give a further background.